This video takes you through the steps to set up a multicolor experiment in the software. Click New Experiment to get started. Type a name for the experiment and click the next arrow. Here, we activate detectors based on the colors that will be used in the experiment. Type the first few letters of the fluorophore name to find it in the optimal fluorophores list. Once you find what you're looking for, double-click on the name to add it to the experiment. Selected colors appear in the visual spectrum graphs in the middle of the screen. These graphs show the emission curve of the selected fluorophores when they are paired with a particular laser. The graphs also show where spillover and other detection channels could be an issue. You can change the name of a parameter to reflect the marker that's being used. Just click in the list on the right and type a new name. When you're ready, click the next arrow. Now select a media type. In this experiment, we're using a rack of 40 tubes. If you intend to use a stat tube for setup steps, click the stat tube add-on button. We're not going to use a stat tube for setup in this experiment. We're going to use the A1 position, but I'll show you how to do it anyway. Click here to go back to the template. Set up the control tubes using the compensation template tool. To include a negative control position, click the negative control toggle button. To create compensation controls, click the compensation toggle button. If you want to use a universal negative control for automatic compensation, select the Use Universal Negative checkbox, but we won't for this experiment. If you need to reorder the compensation control positions, turn on drag and drop. Drag and drop the positions and notice the list over here on the right side of the screen changing. Drag and drop in this video doesn't look as smooth as it does in the actual software, but it's a helpful feature to be aware of. When you're finished, turn off drag and drop. Now that we've set up compensation controls, it's time to designate positions for our experimental samples. In this experiment, we've only included one sample that's fully stained. So click on one unused position. Click the Sample button to designate the position as a sample. Name the sample in the Sample Name field or in the Run List window. Select all of the wells to modify the sampling parameters. Click the Gate checkbox to set a gate limit in the next screen. The volume limit defaults to the maximum for the media type you are using. Be sure to change the volume to reflect what is actually in the tube or well. Activate Return to Sample if you would like the unused sample deposited back in the tube. You can change other sampling parameters such as the flow rate here or temperature control over here, but for this experiment we'll just go with the default values. This next step is optional, but it's nice to know that you can activate pause after the final compensation tube so that the run list will stop after the controls have been acquired, and you can go into the Analysis tab and perform automatic compensation before you come back to the Local Instrument tab and run your fully stained sample. And then once you do that, the fully stained sample FCS file recorded will have the compensation matrix with it, and you can also still adjust your data and analyze your data in third-party software because the FCS files are all exported with FCS 3.1 standards. If needed, activate agitation. Expand the run list to make sure that all the settings were applied as intended. Notice that the stat tube volume is 6. Let's say we set up the stat tube with 500 microliters in the tube, and we'll need to edit the run list. Of course, we're not using a stat tube for this experiment, but I'm just going to show you how to do it. This is where you change the stat tube volume. Here, how the change we just made was applied to the run list. Notice the global and multi buttons on the lower right corner of the screen. Global automatically creates the same plots for all of your samples when you create plots for one sample. Multi assumes you want to set up custom plots for each sample. After the samples have been configured, click the next arrow to move to the settings screen. The gate limit to the compensation controls using the limit field and the setup button in the toolbar. Now create plots for your experimental sample. Create a forward and side scatter area plot for the 488 laser. Next, create plots from the remaining parameters. Make sure to click the Comp button so that your plots are set up to display compensated data. Create a region in your forward and side scatter plot so you can apply a gate limit to your plots. This can always be adjusted later when you start to run sample. Type in a gate limit. Now designate the gate limit as a sample gate limit. You can check here to see if the limit was applied to the plot correctly. You can click in the gate and drag it to a new position if you already have a pretty good idea of where your cell population will appear. To adjust the size and shape of a region, click and drag the anchors. When you are happy with your plot settings and you have loaded your samples on the instrument, click Run. Wait for the run list to load in the workspace. Right now we are running the unstained sample for setup. If you want to run from the stat tube position, click this icon. But we set up our unstained sample in a tube in the rack, so we're going to run from the plate position. The plate position is either plate or rack. So make sure the plate is visible as it is here. Make sure the setup mode is active and click the play button to start running sample. Notice you are running in setup mode instead of acquisition mode. Data accumulated in setup mode will not be saved to the FCS files. Position the gate over the relevant part of your sample population. You may want to adjust the PMT voltages so the data appears correctly in the plot. You can clear the events while you're setting up. If you want to automatically refresh the data, click the Cycle checkbox. You can change the cycle interval by typing a new value in the adjacent field. When you're finished setting up, click Stop. Make sure you've selected the position you want to start from. Select Acquisition. Click the Play button to start acquiring data. The run list will automatically advance to the next sample when the gate limit is reached. Click here to view plot statistics. If you want to manually advance to the next position, click the Sample Advance arrow. You can adjust gates during a run if you need to. Software will continue acquiring samples until the last compensation control has been acquired. Remember, we configured the run list to pause after the last compensation control. A separate FCS file will be saved for each control. In the interest of time, we're skipping to the last compensation control. Notice the template on the left and that a check mark appears on the positions that have been acquired. The fully stained sample has not yet been run and doesn't have a check mark over it. We'll do that after we compensate our controls. Click the Send to Analysis button. Wait for the run list to load in analysis. Here's the data from our unstained control. Here's the data from our first compensation control in the A2 position. Click on the other positions to view the data from the corresponding samples. The PMT voltage is low in this channel and the staining is dim, so it's necessary to move the region to include the stained population, or the positive population. Do the same for each compensation control if necessary. this arrow to define options for auto-compensation. Select Prevent Automatic Region Determination to instruct the software to use the positive population in each control that was defined when you moved the regions. Click the Calculate button to start auto-compensation. The compensated control data appears on screen. Now send the run list, including the compensation that was just calculated, back to the local instrument to continue acquisition. Click this icon. Go to the Local Instrument tab. Select the position in the template from where you would like the instrument to resume acquisition. Here you can view the compensation matrix. Acquisition mode is active, and click Play to acquire data from your fully stained sample. Plots aren't gated to R1. Do that now using the Filter tool. Stained sample data to the Analysis tab. Your data for analysis in third-party software. It can be compressed and include the run list if needed. To save your exported FCS files. Click Home. This takes you to the main Analysis tab. Click the Local Instrument tab. Again, after you have run an experiment, you will see it in the Recent Experiments list on the right. From here you can resume, run, or edit the run list for future use. So now you know how to set up a run list, compensate controls, and run a sample.